I didn't even know that feature existed. That's so nice to know. Only been using Zoom since 2020. So hello, everybody. Thanks so much for coming and also for inviting me to come speak because one, Saskatoon gives me a reason to come have Taco Bell. Two, I love talking about bats. So any opportunity that I get, I will take advantage of. There, okay. So first, I want to start this off by talking about not about my animals, but about a place, because I think one of the reasons why I have been having so much success when it comes to capturing the types of bats I am capable of capturing is because of this place. So I work out in the Cypress Hills. If you are from Saskatchewan, or even if you are not from Saskatchewan, if you have not been, shame on you. It is an unbelievably gorgeous location, and I love it. So beginning in the spring, both hoary and silver-haired bats migrate to the Cypress Hills. Both of these species are considered highly migratory, capable of migrating up to or over, even a little over 1,000 kilometers. It's possible that the hoary bats are coming from as far away as Mexico. And then in addition, I will be talking about the little brown Myotis, which gets a little bit more conservation attention because this species is currently listed as endangered, and it is the most abundant bat that we do get in the Cypress Hills. So Cypress, at least west block where I do my work, there's two different sections, west and center block. Center block is like the more glampy area. They have like a pizza place and ice cream cones. So it's a little bit different, definitely compared to west block. And west block straddles the provincial border between Alberta and Saskatchewan. And it's what I like to warmly refer to as a sky island. And that term I only first heard whenever I started doing work in the southwestern deserts. But whenever I first came to Saskatchewan and went to Cypress, I was like, that's what this is. This is a sky island. It is in the middle of this vast prairie ocean and rises over 400 meters out of the prairies. And it all of a sudden turns into this little pocket of boreal forest. So the species that we get in Cypress are much more similar to what you're going to find hours and hours northward, but right in the middle of the prairies. And so it's like this little refugia for multiple, especially volant migrants that are coming through. Many bats will decide to stay and have their pups in the summertime. And we also have a whole lot of biodiversity there. So it's like this little pocket of biodiversity, and it's incredible there. My two main study species are the hoary and silver-haired bats. These are the two highly migratory species. And they're oftentimes grouped together because they do share some common similarities. They are not considered to be cave bats, but rather tree bats. Arguments can be made for the silver-haired bat, which I would be one to argue, this grouping of them together. But for all intents and purposes, we have been grouping these species because they share so many similarities, such as they, especially hoary bats, almost exclusively roost in trees. Hoary bats usually select things like spruce and pine, where our silver-haired bats are selecting things like, mostly in Cypress, things like aspen, where they will roost and inside of the cavities. Hoary bats are incredibly solitary, where you're only going to find one in a branch with her pups, if it's a reproductive female. Unlike our silver-haired bats that will roost in smaller colonies, we've been finding ones of usually between 10 and 20. So small colonies. And I am personally really interested in this species because one of them in particular, the hoary bat, it's not getting enough conservation concern. And if we don't have more, then there is a very strong likelihood that this species is going to go extinct in as little as 50 years. There have been huge studies with lots of collaborators all across their population range that have done collaborative work to look at population trends. And it's one of this quote in particular that came from this very comprehensive population modeling paper that had some very bleak predictions for what was going to be happening to our migratory bat species. And it is in large part because of the ever increasing wind energy industry, which I will never say is a bad thing. Renewable energy is the direction that we have to go if we're going to have a sustainable future. I just don't think it needs to be at the expense of our bats. These species, the three main migratories, hoaries, silvers, and red bats, are not listed as threatened or endangered. However, that trend is changing. And once policy is put in place, then industry will have more reason to actually do something about it. So similar findings on large spatial scales, regional spatial scales for these population declines. And so it brought me back to Cypress. My first summer here, which was kind of like a practice season, where I was just out at our Cypress Hills field station catching bats for no real research purposes, but we had the permits to do it. And so I wanted to know what I was getting myself into. And I was stunned. I was stunned because I have had, like in my little intro there, like I am from Missouri, which it's so nice to see you, a fellow Missouri Stadian right there. And I have netted bats all across North America, from Missouri to west, to the Northeast, to the Southwest, and now here in Saskatchewan. And nowhere else where I have netted bats can you get reproductive females like you can in the Cypress Hills. I'm sure there's more locations like this, but this is the first place that I have ever heard of where these are females coming to have their pups, rear their young. There's enough resources for these incredibly long-lived, case-selected species to come to. And so I was able to then ask a question, because not only are these bats in abundance out in Cypress Hills, but also Mark Brigham, who I am studying and doing my PhD work, has been studying the bats out there for 20, 30 years. And he has a dynasty across Canada and all of North America of people who he has taught. And so I contacted as many of them as I could to help answer, would I see this same trend on a smaller local scale, and collected a bunch of historical data sets. So this is, of course, I don't know if anyone has met Mark. I know at least a couple of us in this room have. He is the incredibly bright colored shirt right there in the middle. And these are just a handful of examples of some of his students who are now scattered across North America, who traveled all the way back to Regina just to celebrate his retirement. So really, he's a really, really special person. And he's been doing work out in Cypress Hills for such a long time. So taking advantage of that was an obvious thing for me to do, because I wanted to know what is happening with these migratory species out in Cypress. So I collected data from spanning about 20 years, which encompasses this explosion of wind industry across North America. So it's a really great timeline for this type of research. All of the records were collected in practically the same way. The Battle Creek bisects West Block, and we almost exclusively, and this goes back again for 20 some odd years, all of us who have studied there usually catch bats just across the Battle Creek. So these are all of those netting locations are historical netting locations that I have used, and all of these other researchers have used as well. So essentially, it's a more or less standard survey spanning this huge time frame. So I needed to correct for a couple of things, because some people are good at recording data and other people are not. For me, I'm like, you need to write down literally absolutely everything you can. So I had to do a little bit of corrections when it came to looking at abundance trends, including offsetting effort, because some people will net for 5 hours, sunset to sunrise, and others will net for 1 if they don't want to put in the effort. So you have to correct for that. And I used a Bayesian type model to understand the expected change in abundance. So we're going to be talking about proportions and percentages, the likelihood of seeing decreases or increases. So first up, the hoary bat. This is the one that is most likely to be going extinct quite soon. And I found that there was only a 7% probability that we've seen decreases upwards of 50%. So if we're seeing decreases, they're not huge. But then there was only a 15% probability that we were seeing decreases of at least 30% in abundance. So we're still not seeing it. What I did find was that there was, in total, about 81% chance that we are seeing abundance increases of at least 30%. Which is an absolutely terrifying finding to get when you're very aware of all of the literature saying like, hey, this population range wide is actually collapsing. Seeing this was like, how do I publish that? I was very nervous, but also incredibly intrigued. For silver-haired bats, only a 1% probability that we saw a decrease, and a 96% probability that they were increasing by at least 30% or more. So our silver-haired bats are doing an incredible job out in the Cypress Hills. So lots of interesting findings from that paper, that there was no evidence of a decrease in our local abundance of these bats. And in fact, rather, there's a high likelihood that we're seeing a local increase. And if you ever want to go back to this paper or want me to send it to you, I'm happy to send it. Because one thing that's been happening out in Cyprus over the last 20 some odd years is that there hasn't been a lot of fire management. And because there hasn't been a lot of fire management, woody plant encroachment is ever increasing. There are more trees. If there are more trees, there may be more bats. So there's actually a few more explanations that I think on the local scale make a lot more sense as to why we are seeing these abundance changes within these bat populations, at least on the local scale. In which case, I bring it back again to the importance of this place. There might not be a lot of places like this for our migratory bat species. And if we're seeing this local increase in abundance, we might really need to look at what is going on in Cyprus to understand what is so beneficial for these bats to be coming here over and over again. So I kind of went over this. Again, it is one of the known few reproductive locations for these species. It's really supporting these large numbers of these bats. So highlighting the importance of this spot for migratory bat conservation is something that I am like working towards doing and still trying to stress everywhere I can. Because Cyprus, I don't think gets enough credit, and I think it should get more. Which led me to actually my main thesis question. So that was actually all a side project, which thank God I had something to work on during the 2020 pandemic, which that paper is kind of a product of, because during that whole time I had to completely reframe and rethink of what I was doing for my research. Because I'm sure if you started your programs pre-2020, you know how much it affected being able to do research. Imagine being someone who studies bats during COVID. It was difficult to be able to keep doing anything. And so I had collected some samples during my 2019 season, and I kind of had to do science backwards, which was, these are my samples, what can I do? But after going through the process of doing this local abundance paper, it really just got me asking all of these questions. How are these arguably very similar species able to coexist and increasing in abundance if this is such a small area? They're arguably occupying very similar niches. How are they able to do that? So it kind of led me to this question. How are two similar species are able to coexist during such an important time, being reproduction? The bats that come to Cyprus, almost all of them are reproductive females. And that's an incredibly stressful time. And not only that, but these are migratories. So they are migrating and getting into Cyprus at the end of June. And then by the end of July, those pups have to be volant. They have to be flying, and they are gone by the second week of August. So the window of time is this big. So something about Cyprus is allowing these two incredible species to be able to get enough resources, have their pups, and have those pups be volant in time for migration. So some of the samples that I collected over 2019, which I've continued collecting since being able to start up field work again, fur. Taking lots of fur samples from each one of our individuals, along with matched wing photos, along with matched fecal samples. And with those types of samples, I can answer questions such as, are they sharing similar foraging strategies and thus diets? We can get that through our fecal analysis and through wing morphologies, because our wings tell us a lot about how we are capable of flying and what type of flight we are doing. Are hoary and silver-haired bats competing for similar resources? And if they are competing, is there a difference in some ecophysiology measurements, such as our glucocorticoids? It's often equated to being related to stress. However, cortisol is not a one-to-one stress hormone. So if that's something you've heard in the past, not necessarily the case. The cortisol is very much connected to a lot of different organ systems, and having healthy amounts of cortisol in our systems is something that's always going to happen. But there's potential competition happening here, which is an example of an environmental stressor. So is something going to be visible in our cortisol levels? And then finally, do we see differences inter and intraspecifically? How do the species differ between each other? but also how are the species within differing from one another, adults versus juveniles, male versus females, etc. So first, I have extracted a lot of data from wing photos using a program called ImageJ, which you can see me using here. And I just outlined hundreds and hundreds and hundreds of wings, collecting all sorts of different wing morphometric measurements to help me get an idea of not only migratory flight efficiency, but what I was more interested, switching gears to, was foraging mode. The highly migratory species have very long, narrow wings, which makes them much more well adapted for fast, efficient flight. Whereas other species are going to have shorter, broader wings, which helps them maneuver through clutter better. One is easily more able to fly through clutter, such as trees. The other is going to probably crash into something. So we can get an idea of foraging mode and the insects that they can take advantage of based off of our wing morphometrics. Next, I also did a fecal analysis using diet via microscopy, so just picking apart a lot of poop, which I wanted to really highlight my research assistant, Josh, here, because he's really the one, he spearheaded this side of it because he wanted to take it on for his honors project. So he really spearheaded the microscopy of this, and together we attempted doing the DNA analysis ourselves. With our fur, I can get an idea of isotopic signatures, so stable carbon and nitrogen isotopes. I set up a collaborative project with Dr. Bjorn Wissel, who's located in Lyon, France, and prepared all of the samples, but sent them off to him to do the full analysis to get my stable nitrogen and stable carbon sources. And what this can tell you, the stable nitrogen is going to be related to trophic level. And kind of like the lay way that I think about it is it kind of gives you an idea of how beefy your bug is. So because nitrogen is related to, like if you think about, there's a lot of nitrogen in our muscle. So we have our nitrogen scale, which is going to be on our y-axis, and we have our carbon scale. And carbon is going to be deposited differently depending on if you are in a terrestrial or aquatic ecosystem. So it can give you an idea of the breadth of where they are foraging. So are they foraging on a lot of aquatic and terrestrial insects, mostly aquatic, mostly terrestrial? We can get a broad idea. Yeah, and that's going to be on our x-axis. And you can form an ellipses around that to get an idea of their isotopic niche. Then I set up another collaborative project. If there's one thing that the pandemic definitely taught me is that collaboration is such an incredible tool that I'm not sure enough people take advantage of. But being able to work with other scientists and being able to work with other labs where I didn't necessarily have a lot of expertise, but knew I could answer cool questions if we worked together, has led to so many amazing collaborations and has made my personal PhD experience just incredibly rewarding. So I first found this paper, which was great because I was like, oh, this is almost exactly what I wanted to do. So I reached out to the first author and she directed me to Dr. Gabriella Mastromonaco, who just so happened to be at the Toronto Zoo. So I set up a collaboration with her and the zoo, and we've been measuring for cortisol levels. And with that, there is a small assumption because there's not a lot of research on bats. There's still so much we don't know. And we know a lot for like this many species, and we know this much for this many species. And my migratories are one of those, we know this much. And some of that goes into fur growth because everything, we shed all the time. So do our bats. But there is a single study that was able to give an idea of how these bats are growing fur and when they have their molt. And it was supported that they usually go through a molt once a year, right before the pupping season, which I was like, great. Because that means the fur that I am likely collecting in Cyprus is fur that was grown right there, right then. This is going to be especially for the juveniles that I've sampled, because that means that all of the fur that they grew was grown in Cyprus. So some assumptions about fur growth, but overall I have a pretty good idea that the fur was grown prior to my sample collection. For wing morphometrics and foraging ecology, the two main things that I wanted to look at were aspect ratio and wing loading. So aspect ratio is going to be the wingspan divided by the wing area. And again, it's for our migratory species, more highly migratory species that are going to have longer, narrower wings. They will have a higher aspect ratio. So means higher aspect ratio means long and narrow, lower. Oh gosh, that is, oh, right. And higher it still means also flying efficiently and for longer. I predicted that hoary bats, of course, would have higher aspect ratios. This has been supported time and time again, but due diligence, I wanted to measure my own bats. So supported. For our wing loading, that is related to the mean pressure on the wings, so how much force we can get. And that gives us an indication of faster flight. Higher wing loading, faster flight. Well, if we are a highly migratory species with those nice long narrow wings, likely we're going to have our higher wing loading as well. That is supported yet again. And coming up very soon, I have started, the little brown Myotis was added into my thesis because of increased sample collection for the last 2 years. So I'm almost done with the outlining of wings, because again, there was probably like 500 pictures to go through. So a lot. Yeah. And so we have long and slender for our hoary bats below here versus a little bit shorter and broader for our silver-haired bats. And if little browns, which are soon to be included, they would be even lower in value and have even shorter and broader wings than either one of these two species. For our dietary findings, again, highlighting Josh here because he put in so much work, we had to rely on microscopy alone for now, because unfortunately, as much as Josh and I tried, we only had so many fecal samples, and we attempted 4 times to do our DNA extractions, and 4 times we failed. So we decided, let's do microscopy on what we have remaining from those samples. And then we sent off the rest of our samples to the Species from Feces Lab in Flagstaff, Arizona, for more detailed and much more professionally done DNA extractions. I did just get those results. And in fact, my goal was to complete including updated figures, but my computer wasn't working in the car and I couldn't do that. For all intents and purposes, this holds up for our silver-haired Lano, which stands for Lasionycteris noctivagans, and Lasiurus cinereus. Their diets are incredibly similar. Beetles and moths are taking the top shelf when it comes to our migratory species. And if you were to see these bats, you would kind of understand, because they have massive, massive teeth, especially the hoary bats have incredibly strong bite force. And that's in large part because one of their primary prey is these very juicy beetles, which I can now confidently say are ground beetles. Yes, so stay tuned for the fall when I have like my full PhD analysis completed. But I finally have the diet analysis, in some cases all of them going down to family, and for some of them going down even to genus, which is really exciting for me to start diving into, especially for our migratories. When it comes to our isotopic niches, I did want to kind of quote this paper here, because I think it's really important to stress that the niches that I am looking at are the isotopic niches, again, the nitrogen to carbon relations to one another, and they can be incredibly comparable to the ecological niche. Very, very comparable, but not one-to-one. So it's going to give us a good idea of how they are interacting when it comes to foraging in the ecosystem, but not the full picture. So for this plot, all the individuals are the points inside, and the ellipses kind of encompass the entire area of what that isotopic niche is going to be. We have our stable nitrogen isotopes on our Y again. So again, that's related to trophic level. And then we have our carbon scale on our X, which is going to be related to where in the ecosystem, terrestrial versus aquatic. And our hoary bats are the large black circle, and our silver-haired bats are the red nested right in the middle. And whenever I first saw this result, I was incredibly intrigued, because to me, it looks like there's potential like complete overlap of these isotopic niches. But then when I think about it, what we see is that the hoary bat has much larger scales on both the nitrogen and the carbon scale, which might be an indication that their diet is much more diverse when compared to our silver-haired bats, because they have more breadth on where they're foraging, and they have more breadth on the potential meatiness of the insects that they are foraging on. So I'm really excited to be continuing to update this. I should be getting 2 more years worth of results for this. I keep saying, I bet it's going to be next week. So I bet it'll be next week. We'll see. So this is for our adults. And next I'm going to show juveniles, which something to consider with the juvenile plot is that the juveniles up until this point have been foraging on their mom. So again, this is based off of fur. So the fur that they grew while nursing. And so what I would expect is to see something that looks rather similar to this, if not even more reduced, because all of the isotopes that would be stored in the fur of the juveniles would have been produced based off of what the moms were probably eating themselves, which I find something incredibly what I practically predicted I would get. We still, if you kind of go back and forth a little bit, the hoary bats are still very much more broader on both the nitrogen and carbon. And then our silver-haired bats are a little bit more nested within that. So this is pretty much what I expected to get. And I'm really excited to also expand on my juveniles as well, because at this point, the sample sizes are getting a little too low for comfort. And I would like to piece it apart more, but not until I have that sample size increased. When it comes to cortisol levels, so again, this is going to be like a physiological response to the environment. This is based off fur cortisol, and I do want to point out that when it comes to how you collect your samples, cortisol is going to be deposited differently. You can get cortisol from blood, urine, feces, and fur. Fur is going to be prolonged deposition of cortisol. So this is long-term cortisol levels because it takes time to grow fur. So this is a long-term look at their cortisol levels versus blood and urine is going to be very quick pace. This is what, if anyone has ever collected or has been looking at cortisol samples before, you have to be very quick with it if you're doing blood cortisol, because it can change so rapidly and get circulated into the body. If you stress the animal out too long and you're not fast enough to get your blood, you might have artificially spiked that cortisol level by accident. So this, don't have to worry about that. This is all from fur. And so these are all prolonged levels of cortisol. And there is a statistical difference here between our species. So we have our hoary bats on the left and our silver-haired bats on the right. And when it comes to the amount of cortisol, our silver-haired bats were slightly elevated when compared to our hoaries. And so I wanted to dive a little bit deeper. And I separated it out by age class. So we have adults, both adult species on the left and the juveniles of each species on the right. And when I look at this, and again, our sample sizes are getting a little bit smaller here, so I have to take it with a grain of salt, but our juvenile silver-haired bats are what was driving everything up. When you remove the juvenile silver-haired bats, everything else was practically no statistical differences. So something is happening with our juveniles. So looking at differences between sexes, we see that there was no statistical differences here, even though we do see a lot of variation within our female silver-hairs. And again, I'm excited and curious to see what happens whenever this sample size starts going up. I want to know if these trends are continuing, or maybe even if we see differences between years. Maybe something in the environment is changing between, this was 2019. So what happened in 2021 that is going to change this? And that, again, is soon to come. Can't wait. Looking just within species, our hoary bats, cortisol by age, and this is what I would expect. Because again, the juveniles in this case would have been nursing from the moms. And so the cortisol deposited in their fur would have been taking a lot of that from the mom directly. And with our hoary bats, our juveniles are smacked out. They are practically the meat, which you can kind of see here. And so then, just looking at our silver-haired bats, it makes it even more dramatic to me to be looking at it this way, where our adults are significantly lower than our juveniles. So something that the juvenile silver-haired bat specifically is experiencing in the environment is causing these elevated levels. So we'll see if this holds up between years as well. And if it does, like, that's just going to open up this like very strange box of questions for me. Yeah. So really interesting results here. I don't currently have a good explanation as to why this would be. What is it that these juveniles would have been experiencing to cause this spike? So I wanted to know also if size matters. Okay, so when it comes to our adults, there was no relation. It did not matter if you were a small or large silver-haired bat, did not matter if you were a small or large hoary bat. There was no trends whatsoever when it came to whether or not size influenced the amount of cortisol level. But if you're a juvenile, it makes a big difference. The bigger the bat, the bigger the pup, the lower those cortisol levels were. So again, maybe there's something about juvenile interactions within the environment where there's more competition just between juveniles. Don't know yet. So again, still am piecing apart, and I want to wait until I get all of the results for further cortisol analysis before I start postulating too many things, because this could just have been a weird year. And until I start like really diving into those new 2 years worth of data, I won't know. So when it comes to summary to date, the wing morphologies suggest that they are likely going to be having differing types of foraging strategies, where hoary bats are going to be more capable of being in more open areas. Our silver-haired bats are likely able to have a little bit more maneuverability within that clutter. But our diets and isotopic niches are incredibly similar. So now that I have my full diet analysis done, I'm going to start really piecing apart what are the main differences between which beetles they are eating and between which moths they are eating, because that's where we might see a lot of separation between their diets and how they're able to coexist so well with one another. And the juvenile silver-haired bats seem to be more physiologically susceptible to the environment. Still an unknown reason as to why, but I am excited to see where those results are going to go and how those results are going to grow. So up until this point, I've been focusing mostly on my two migratories, the hoary and the silver-haired bat. And now I'm going to bring it into a species that's considered a regional migrant. So the little brown Myotis, Myotis lucifugus, is considered to not necessarily, like, they just don't think that little brown bats are super migratory, but they can move up to like upwards of 500 kilometers to get from a hibernacula to their reproductive grounds. Cypress, yet again, is an outstanding place, and there is an incredible maternity colony of little brown Myotis located, of all places, right at the ranger station. So we can just set up our, like, there's chairs and tables and horses hang out with us, and then we just get to catch bats outside of a barn. It's fantastic. And they got thrown in to my thesis because I started to think about how it's really just these three species that are in Cypress. There are big brown bats, which if you live in Saskatoon, you've probably like seen them flying around at night because they are literally everywhere. If you have bats in your house, they're probably big browns. If there's bats in a church, it's probably big browns. There's bats in the U of S. They are likely big brown bats. They're everywhere. But for some reason, out in Cypress, they are the least frequently caught species. Don't know why. So it's mostly these three. So why not? This is still a migratory species, not as extreme as the other two, but we'd be able to draw some like really interesting comparisons between these different migratory methods. So the little brown bat is an endangered species in Canada, about to be listed in the States as well. We don't know a lot about them in Saskatchewan. There's a lot of resources right now that are going to little brown research because white nose syndrome is the fungal disease is wiping out, decimating populations across North America. And it did hit Saskatchewan a couple of years ago. Thank God we have not gotten any confirmation that any of the samples we've sent in from Cypress have come back for positive of the fungus. So we're very happy about that, but we don't know where they go. We know that they come to Cypress. We have no idea where they hibernate. Nobody does. So there's a lot of effort going into trying to understand them. We don't know the full extent of how they're impacted by white nose syndrome. And we don't know a lot about their ideal habitats in Saskatchewan. So luckily for us, we have the resources out in Cypress to be able to study this species as well. So this is an ongoing research project that kind of spurred me wanting to include the little brown Myotis into my thesis. It started with a collaborative project with all sorts of different entities. And it really all started because I found a common connection between salmon and bats. And the answer is this man. This is Dr. Kurt Samways. He is a professor at the University of New Brunswick and per usual is also part of the Brigham dynasty across Canada. He started his program at the U of R. He's originally from Regina and is now one of the top salmon migration biologists in the Northeast. So he's absolutely incredible. And he came to do a seminar at the university. And we were just sitting around the table, kind of just shooting ideas across one another. And I was like, well, you know, it's so interesting. Like the way that you study salmon, he sets up these massive big antennas and the salmon pass through them and they monitor them. And I was like, that's such a great way to use this like three-dimensional medium to understand how these fish are moving across within the creeks. And then I thought to myself, well, the bats use the creek exactly the way fish do, like a little highway, and it's a three-dimensional medium. So what if we put your antennas just over the water instead? And we wrote a grant together and it got funded by the federal government for the tune of $100,000+ So we did it. Is a large-scale passive monitoring system that might be a novel way of monitoring bat movement. And a lot of the times when people monitor bat movement, it's in relation to wanting to find roosting locations, or they will monitor movement using things like acoustics, but not real time. This bat went from here to there in the context of foraging and how they might use waterways as like almost like little highways to access different foraging areas. So we wrote up that grant and started implementing this project in 2021 with this massive antenna system. So here's just a couple of pictures from this project. It was incredibly intensive to set up, and we know it works because we've released bats nearby and they pass through and it pings. However, we have not gotten what I like to call a natural detection yet. And part of me, I'm actually going to be optimizing this over the next couple of weeks because part of me thinks that our bats just move too fast. And there's not enough time passing in through the antenna and for that signal to be sent to the antenna, just too fast. But we are using pit tags. And we are pit tagging every single bat that we can possibly pit tag. That includes all of our little browns, all of our hoaries, and all of our silvers, so that we're going to get an idea of how they are moving, not just foraging as a species, but also potentially interacting with one another in that ecosystem. So here, up to date, we have tagged well over, I think, 100+ little brown bats, 80+ hoary bats, and 100+ silver-haired bats. What's really interesting about my time out in Cypress as well is that, you know, I'm out there practically every summer catching bats as much as I can. And I have never gotten a recapture of any bat that I have captured, not a single one. And we're talking hundreds and hundreds of bats at this point. That is not the case, though, for a bat that was caught about 29 years ago. One really, really cool finding that we had, and another credit to Cypress, is that this is a location where bats are coming to decades later. This was a really exciting find because we are getting recaptures. They're just not the ones that I personally caught. This little brown Myotis was originally caught in 1993, and my team and I caught it again 28 years later, and it was already an adult, so it was at least 29 years old. So this is, again, yet such an incredible location to study bats and bat foraging and bat migration. It's just, there's so much that it has to offer. Yeah, so this is also an incredible location for our little brown Myotis as well. And it's kind of in addition to, this has happened before, where in Cyprus, we caught a bat. This was a part of the team where Shelby was working on her master's project and caught a 23-year-old bat. So this is a reoccurring thing happening out in the Cypress Hills. And it also connects us with the original students that Mark has taught. Dr. Martina Kalkunas-Ruppel is now Dean of Science over at the U of A. She was the original person that caught this bat. She was the original person that caught that bat, all from the early 90s, which is just such a neat connection to make across time. So why is all this important? I think you can probably, I think I've kind of made the case already. What is going to be happening on our summering grounds, especially during the summer pupping season, is directly going to influence migration survival. Migration is incredibly risky. This is why my migratory species typically have twins. because migration's risky. Better to have two. But they have enough resources in Cyprus. And again, this much time to be able to accumulate all of the fat that they're going to need for migration and potentially hibernation. It's not one or the other, especially for our little browns. They're migrating up to 500 kilometers and then hibernating. So they have to be able to get enough resources, to get enough fat stores to move that far and to survive the winter. Cyprus is potentially a place with an abundant resources, which is critical for our migratory bat species, and aerial insectivores, like that's including all of our birds as well. Aerial insectivores are drastically declining. It is a problem across the board. And so having this essential ecological and biological knowledge is going to help further conservation efforts in the future. As these species likely continue to decline, we have to know what they need in order to help the species survive. Otherwise, we are not going to see them for much longer, and we will potentially within our lifetimes see species as incredible and beautiful as the hoary bat literally disappear into the darkness, never to be seen again. With that, I have way too many acknowledgements. This is us in the Brigham Lab having fun out at the field station. And if you ever want to contact our lab and spend some time out at the field station, highly recommend it. It's an incredible place filled with incredible and brilliant people, and it will be a good time. So yeah, thank you for everything. And again,
Thank you, Dana. That was awesome. Does anyone have questions? Hop into the microphone because then people on Zoom hear you. Yeah.
Okay, hi. I just have a question about your mist netting actually. And I was wondering, like, with those really high nets, how do you bring it down when there's like a bat in the top row? I can bring this up.
Or we can just have you repeat the question.
that's true. That's a good idea. Okay, so I'll just repeat so the question was, in case no, you had the mic. I don't need to repeat it. So that is a triple high pole set, and it's a pulley system. So I'll see if I can find a picture. Okay, while I'm going, anyway, it is a pulley system. And so the way that the nets stack, there are 3 or 4 bars that we all hook together. And at the very end, so there's distinct sides. There's definitely a top and there's definitely a bottom. And I've always called them a changer set. The different loops are called changers. And you hoist them all the way up to the top when you're putting it away at night, and it groups them all at the top. And then once you're ready to let them down, there's just one big rope and you just release it and they all come crashing down. So when it's time to raise the net, you raise them starting with the top one first, and you put them on in like opposite order. So top goes first, then the middle, then the bottom. And as you're hooking them onto the changers, which have carabiners hooked onto them, you hook them onto the carabiner, hoist it up, hook it on, hoist it up. And so it's like a big pulley that goes all the way up to the top. But yeah, triple high pole sets are the way to go, for sure. Huge pain in the ass, though. I don't know why this presentation's not closing now. Yeah, here's a good picture of it. So each one of those loops is called a changer. And it takes some practice getting used to this, but they're really not difficult to understand. So there's a lot of different poles and you can actually use these sets. You can use them as a triple high set, or you can use them as a double, or you can use them as a single, which is why I like them. And I've actually kind of stolen how they've put this together and made a couple of homebrew pole sets myself because these are expensive and research funding is limited. But I won't sell them, so I won't get in trouble. But I'm happy to share the design. Yeah.
So how often do you get to do this?
So the question is, how often do I like check the nets and make sure there's no bats in there? There's a really one big time frame where we get a lot of bats, and that's right around emergence time. So right as the sun goes down, all of the bats are coming out at night, and that's when we're going to get the most action. So if it's a busy night, we'll just have to stay at the net because we're constantly catching bats. But after a while, it will die down over time. And we will check the nets depending on how busy it is, either every 10 minutes or every 15 minutes, but usually it's every 10.
And so if you catch one in the top net, you can drop that down?
Yeah. Yeah. Sometimes you will get bats like all the way at the top and all the way at the bottom. And the only thing you can do is get the bottom one first and then lower it down and get the top one. Mm-hmm. Did you have another question? Okay. Mm-hmm. To find hibernacula sites, probably not. At least not in the way that we have it set up, because this is very much focused on foraging ecology. We wanted to understand how they are moving between clutter or open areas to see if they're more selective of one or the other, depending on species. But to get to hibernacula, bats are moving hundreds of kilometers between areas like this to get to a hibernacula. So there is a lot of funding and research currently going into setting up the large-scale Myotis project. Who is not familiar with Myotis? Everyone's familiar with Myotis. I'll tell you about it later. So there is a lot more Myotis towers that are about to be going up to specifically, like I know that like there's a lot of obviously bird people will use Myotis. Like Myotis is huge for bird research, and it's only just now starting to be implemented for our bats. So they're multi-purpose too. Pretty much anything that flies, you can use Myotis towers for that. And there's a number of bat labs across the prairies right now that are all co-collaborating with the federal government to get more Myotis towers put up, including I think Last Mountain Lake is going to be getting a Myotis tower as well. So a lot more monitoring to find out where they are going for hibernacula, because we don't know. But what you could do, it doesn't really work well because of many physics reasons why. Having an antenna outside of a hibernacula, like putting one of those big ones around a hibernacula to see if you could ping all the individuals coming in and out, doesn't work because two tags can't be read at the same time. So if you have like hundreds of tagged bats all flying out of your hibernacula at the same time, it's not going to capture it. For physics reasons, but we can talk about that later, but physics is the answer. Oh, sure. Okay. What is known about demographic rates, juvenile survival? I wish I could tell you. I wish I could tell you. That's a great question. It's a question I would love to know the answer to. But again, it's kind of one of those, we know very little about too many bat species, including this one, including this question. So I wish I could tell you. The answer is I don't know. To Kevin. Sorry, Kevin. Michael, will this presentation be following? Wait. Yes? Oh. Would the flyway monitoring potentially be something that could be done with citizen? Yes, but no. With this particular setup, there's a little bit too much high voltage equipment that is involved for me to feel comfortable putting a citizen in charge of that. But there's lots of citizen science projects monitoring bats and bat activity using acoustic recorders. We actually did a big study across Regina where we got a bunch of local citizen scientists to have bat recorders in their backyards so that we could understand how the pandemic influenced bat activity across the city. So there's different ways to involve citizen scientists. Working with high voltage electrical systems, I don't think is one of them. Yes, on the YouTube question? Yes, on the YouTube question. Thank you. Oh, hey, Jeff, nice to see you. Quick question, demographic population results. Cyprus is pretty geographically isolated place. How reflective do you think your results are? Population growth, continental patterns. Could it be that there are different migratory pathways for, I don't know why that keeps changing, sorry. My personal feeling on it, and with my understanding of population demographics of migratory bat species, that this is a local abundance phenomenon, and that it is something happening in Cyprus that is causing them to have this kind of localized abundance increase. So how reflective do I think my results are of population growth? I don't think they are. I don't think that they're reflective of overall population growth. I think it is purely reflective of local abundance trends. That is my interpretation of what I found. But we can definitely talk about that more. I do expect to see you at NASBR. So could it be that they're on a different migratory pathway, for example? Entirely possible. Entirely possible. We don't know a lot about migratory pathways of these bat species yet. Again, I wish we knew more. There are more and more newer research papers coming up doing predictive modeling for migratory pathways, but it's still kind of one of those we don't quite know enough. But one interesting thing that I think, Jeff, you would find particularly interesting is that something about last summer. Very rarely have we caught red bats in Cyprus. They're here, they're in Saskatchewan, but over the last 10 years, I think 1 or 2 maybe have been caught in Cyprus. Last year, they caught 3 in like 4 weeks, and I wasn't there, and I was so mad. So I don't know. It could very well be that they're different migratory pathways. Again, it's one of those I wish we knew more kind of questions. How far are these species typically flying from their roosts when they encounter your nets or antennas? I'm going to back up to a map real quick. It does depend on species, how far they're going to be going. But I did do my first field season was going to be like I was going to be tracking a lot of silver-haired bats to roosts and climbing those roosts to get all sorts of different demographics. However, when one goes half blind right before a first field season, going very high up in trees becomes something that you may not want to do. So I did not do any roost tracking, but we do have Emma in our lab who is tracking little brown Myotis bats and to their roosts, and they're not going too far. The females in particular all seem to be coalescing on this one massive maternity colony, which has hundreds of individuals. And otherwise, they're kind of just selecting scattered trees across the landscape. But for some reason, they seem to be focusing more on spruce trees rather than our aspen trees, which our silver-haired bats like to go to. But since you're online, for example, I caught silver-haired bats here, and they were roosting in here, which is incredibly close to our field station, which was lovely for me because then tracking was super quick and I got to go home. Well, to the field station, same. How far are these species typically flying from? Yeah, so I would say within Cypress Hills, for the ones that are roosting there, fewer than 10 kilometers away. So they're roosting very locally. If they are roosting there, we have had bats just leave. So it happens. Some are transient because these are migratory species, and there's equally a sexy boreal, a couple hours north of where Cyprus is. So if there is not enough available habitat for them to feel like they are capable of going through all the motions of surviving the summer, then my assumption would be is that they continue their migration and just use Cyprus as a stopover site, in which case it's still incredibly important for migratory bats because stopover sites is a whole other thing that I could go on a rant about. But yeah, any other questions? Yes.
So you're saying that there's an increase likely of the bats because there's less fire. So would you say if there was prescribed fire projects in the area that it would negatively impact the bat population?
At this point, I don't think so. At this point, the forest has been, it is historically, I think would now be considered overgrown forest. I actually, every time I go out to Cypress and I'm about to spend the summer there, a part of me is in a constant state of anxiety because it will catch on fire. It is a tinderbox waiting for a spark. We actually got evacuated for almost a full week because there was a fire, not even in the park. It was north in a coulee somewhere. And they were so worried that a spark was going to come up because when a fire starts in Cypress, it's going to be huge. Unless, they do some very needed fire management. I would encourage it. And at this point, these trees are very well grown. And so there's lots of available fully grown trees, whether they're spruce or aspen. So I don't think so. Probably for a short time, I would of course expect to see some differences if there was a fire, but also what a great opportunity for research because it's coming.
And then my second question was, times for the bat or even birds as well?
Right now, for migratory species, they're not protected. So, do you think there's mitigation? Yeah. No, there's not a lot. I know that I think a lot of industry individuals are starting to really prepare because the listing of these species is inevitable. It is coming. And so at this point, it will behoove them to set some mitigation standards across the board. I don't know what those are going to look like yet, especially for migratory animals that have such a complex set of requirements as they go along that migration. I don't know which part are you going to mitigate for? So I'm interested to see what happens and hope to be a part of the conversation as well. Yeah.
I was listening, but is there something about bats like patterns that makes them more at risk for turbine-related deaths and injuries than other migratory species?
It depends on which one. It really sucks being someone who studies migratory bats because each one of them, even though they're arguably very similar and some of them closely related, they behave in such crazy different ways. Red bats, for example, are one of these top 3 species that gets knocked by wind turbines. Second, so it goes hoaries, red bats, and silver-hairs. We just don't get enough red bats here for me to really get a good idea of what's going on with them. Red bats, you can use acoustic deterrence. They respond to that. You can put up acoustic deterrence and it would actually lower the risk of finding dead bats under your turbines. Doesn't work for anything else, just red bats. For hoary bats, and we do not know why, hoary bats seem to be attracted to wind farms. So we don't know what's going on. We don't know why they're attracted to it. There's lots of research that's currently going on to understand what is it about these turbines that is attracting them to this area. This is the most tree roosting species out of these ones. And so is it something about the turbines that is is there something alluring about a big tree across the landscape? We don't know. And what's even worse is that it doesn't necessarily, and this is across the board for bats, it doesn't need to be a collision for them to experience mortality. A lot of the times what they're finding is that if you think about, if anyone's ever been next to a big wind turbine before, it's almost like you can feel it. You can almost feel it like a pressure change because there is one. This is a giant blade that doesn't look like it's spinning so fast, but it's moving so fast that the tip speed of those blades is crazy. And what that does is create this massive pressure change right around the turbine, right around those blades. So the bat only has to get close enough to increase this massive air pressure change, and it causes barotrauma inside of the inner ear. And so the ear practically bursts. And if that doesn't kill them immediately, it disorients them enough that they end up experiencing mortality for another XYZ reason. Yeah. I know I saw a question back here. I definitely wouldn't think that it would hurt. Whether or not it's directly helped, I have no idea. And I'm not sure of a good way how to measure that either, to be able to answer that type of question. It's a good one because there's not a lot of these dark sky locations. Yeah. 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 It depends on if they have a population like the local one in Cyprus. Yeah. Great question though, because there's not a lot of dark sky preserves out there, which again, another reason to go to Cyprus. The skies are incredible. Yeah, just like shoot me an email, come out to the field station. We'll be catching bats there all summer. Oh, okay. Darn it. You mean you do field work? What? Let's see if there's. Oh, okay. Good. Okay.